This last video talks about some of the applications or ways that we use and modify DNA in a laboratory. Now, almost all of these techniques go back to the structure of the DNA molecule and the fact that that molecule is complementary, that the sequence of base pairs has a specific complement to it. So the overall structure and this idea of complementary base pairing has been used for a variety of purposes. So we're going to talk about a couple of them here. In our upcoming lab, we're actually going to do a little bit of restriction analysis. And we're not going to do any of the others, but they are quite interesting. And I definitely cannot go into detail on any of these. Some of these would require an entire hour-long lecture on their own. So I won't go into all the details here, but I will give you a general overview of some of these techniques that are used in genetic engineering. So the first is using something called a restriction enzyme to make recombinant DNA. So let me tell you what those words mean. A restriction enzyme is an enzyme that was originally isolated, or I should say it's a group of enzymes that were originally isolated from bacteria. Now they were actually sort of a bacterial immune system that recognizes infection by viruses or other foreign pieces of DNA. And restriction enzymes, they're actually called restriction endonucleases in bacteria, will cut DNA at very specific locations. So they're a natural defense mechanism. They're used to break down DNA, but they were discovered and then isolated, and they've been really, really handy in molecular biology techniques. So as I said, they recognize specific sites. These are called restriction sites, and cut DNA into restriction fragments. So each restriction enzyme recognizes a very specific fragment, and it always recognizes the same fragment. Those fragments are palindromes, which means they're the same forward and backward. So on one strand, they have one sequence, and on the other strand, they have the opposite and backwards sequence. So this one in the image here is called EcoR1. Eco meaning that it was taken out of E. coli, R1 just identifying it as one of the eco enzymes. You'll see that it cuts at the sequence GAATTC, and if you look at both strands, you'll see that one opposite of the other on the complementary strand. Now it cuts in a configuration that produces what we call a sticky end. Remember that the DNA strands are complementary. So if you actually cut two pieces of DNA with the same restriction enzyme, you can use this principle of complementary base pairing to stick them together. Now you can see that in this image we have inserted that pink piece of DNA into the green piece of DNA, and we have produced what is called a recombinant DNA molecule. Recombinant, you may remember that word from chapter 12 when we were talking about the chromosomes that are shuffled through crossing over, and we get these recombinant phenotypes or new combinations of phenotypes between the parents. It's sort of the same idea here. A recombinant DNA molecule is a DNA molecule that has information from two different sources. So that green one is actually a little tiny specialized chromosome in a bacterium called a plasmid. And so we cut that plasmid at a specific site, and then we cut in or stick in a gene from somewhere else. So this might be a gene for firefly luciferase. That's a gene that causes the firefly abdomen to glow. And so you can insert that gene into a plasmid, and then you can make your bacteria grow. It's actually really cool. That's one of the labs that we do in genetics and in Biology 101. But anyway, we now have a molecule that is made up of both bacterial DNA and the DNA from another organism. One of the techniques that we use to analyze DNA is called gel electrophoresis. So we can take a sample of DNA, cut it up using restriction enzymes to isolate the piece that we want, or we can just run it without cutting it, just see what we have. But DNA electrophoresis allows us to separate and visualize fragments of DNA in a gel matrix. So this is sort of like a really thick jello, and you load your DNA sample into one side where there are specific wells, and you load each DNA sample into their own well. Then you hook the your DNA gel up to a current, and you allow the DNA to move towards the opposite end based on the current. So one thing you might remember is that the phosphate group has a lot of oxygens, and that makes the DNA molecule overall negatively charged. So when you apply a current to it, it pulls the DNA towards the positive anode in your electrophoresis chamber. These DNA molecules then separate by size, the smaller ones moving faster than the larger ones. And so you use the charge to separate the DNA fragments, and you can visualize them very nicely. There's a picture there that is showing a restriction digest. The smaller molecules move farther down the gel than the larger ones. And that just allows you to analyze different variations in DNA sequences. So let's say that you want to take a gene out of one organism and put it into a bacterial plasmid. You want to make a piece of recombinant DNA. You likely have a lot of plasmids because you've grown a lot of E. coli, and they contain a lot of plasmids. And so you're going to need a lot of copies of that gene that you want. So the first thing that you're going to do is you're going to take genomic DNA from whatever organism you want. Let's use our firefly. And you're going to need to cut that gene out. So you would isolate the DNA, you would extract the DNA and separate it from the other cellular components, and then you would do a restriction digest on it. You would cut out the gene that you want, and then you're going to use a process called polymerase chain reaction. Polymerase chain reaction is essentially DNA replication in a bottle, in a machine, and it allows you to make a whole bunch of copies of a very specific sequence in your DNA. Usually it's a short sequence, not longer than a single gene or so. Quite often it's even shorter than that for different purposes. But in this process, you are going to use heat to separate your DNA molecules. Then you are going to add a primer molecule. So instead of using RNA polymerase to put in a primer for you, you're just going to add a pre-made primer, and it's going to complementarily pair with your genomic DNA, surrounding the sequence that you want. You're going to allow those primers to anneal or to stick to your DNA. And then you're going to add some nucleotides, and you're going to add a DNA polymerase. The most common one is called Taq polymerase. It's called that because it was isolated from a bacterium called Thermus aquaticus, that it was isolated from a bacteria that lives in hot springs, so it can withstand really high temperatures. So you're going to allow the DNA polymerase to replicate your parent strands. You're going to do this over and over again until you have isolated the gene that you want, and you've made lots and lots of copies of it. Every time you perform a cycle of PCR, you double the amount of DNA you have. So in theory, you can start with one copy and get thousands of copies. So after that, you have your DNA fragment that you have produced or copied via PCR. Both your bacterial plasmid and your PCR DNA have sticky ends from restriction digest. And you are going to mix them together and allow them to stick to one another. And eventually you have what we call a cloning vector, and you have made it into a recombinant DNA plasmid. So a cloning vector just means a piece of vehicle, basically, for inserting your DNA that you want. And then you're going to put this back into the bacteria that it came from. And when you do this, you're going to allow that bacteria to express the gene that you're interested in. So let's look at what's going on here. So we have a bacterium at the top. Let's call this E. coli. We remove the plasmid. We cut it with our restriction enzyme. We put in our piece of DNA that we're interested in. We mix it together. We let them come together in a test tube. And then we use a process called transformation, which is just the process that puts the plasmid DNA back into the bacterial cell. And the reason that you do this is that it allows the bacteria to clone or copy that plasmid. It also allows that bacteria to produce the protein that's going to come out of that gene sequence. So we let the bacteria do the dirty work for us. So I said a minute ago that maybe we wanted to make our bacteria glow, so we're going to use that firefly luciferase enzyme. We would insert it into the plasmid, and as long as we have all the parts of that gene, we can use the rest of the plasmid and the molecular machinery of that E. coli to produce the proteins that we want. This is used in a lot of different ways. There are some examples here. One is inserting genes for pest resistance into crops. Another is for inserting genes into bacteria that allow them to break down toxic waste. We can also use this for medical purposes. So one way that this is used is to grow human growth hormone. So this is used for individuals that have stunted growth for a particular reason. We allow a bacterium or some other organism to grow that and produce that human growth hormone, and then we can isolate it and give it like a medicine. You can also build proteins that dissolve blood clots. These are used in heart attack therapies, and the list goes on and on and on. There's a ton of things that you can do with DNA cloning. I'm not going to talk too much about the process of DNA sequencing. DNA sequencing is essentially PCR with labeled nucleotides, and those labeled nucleotides are read by a computer program that will tell you the sequences of the base pairs. There's lots of different forms of DNA sequencing. Now it's very automated, very fast. You can get a lot of sequence information very quickly. It didn't used to be that way, but DNA sequencing really took off with the Human Genome Project, which was quote unquote completed in 2000, but it was made possible by some advances in DNA sequencing techniques that made it faster. And in the past 20 years, it's gotten even faster and more accurate. All right, so let's get into the cool one. The most recent development that we talk about in our book is something called the Crispr system. So the Crispr system is another bacterial immune system type protein. Basically what it does is the Crispr system along with some other mechanisms in the cell, insert pieces of foreign DNA. So it's sort of so they use restriction enzymes to cut up invading DNA, and then it actually inserts little chunks of that foreign DNA into the genome of the bacterium in this CRISPR region. And in this CRISPR region, it's sort of like our immune system where we have antibodies built up to a vaccination. It allows for the bacterial cell to remember that foreign DNA so that if that foreign DNA is infected again, it can establish essentially an immune response and break it up even quicker. So the CRISPR-Cas9 system was discovered, and it's been more recently used to introduce mutations or correct mutations in bacterial cells. Now, the reason that it can do this is it has a short segment of RNA that recognizes the sequences that had been put into the bacterial genome. So it has basically a complementary sequence to the foreign DNA. It recognizes the foreign DNA and then it cuts it up so that it cannot infect the cell. That process has been utilized to actually edit genomes within all kinds of organisms. So this diagram shows what's happening in the CRISPR-Cas system. A bacteriophage injects some DNA. That DNA gets broken up and is inserted into the CRISPR region. Then that CRISPR region is copied, turned into RNA. We'll talk about that process a little bit later. And along with a set of proteins called the Cas proteins, that RNA will target DNA the next time that bacteriophage invades. When it recognizes this foreign DNA, it makes a cut. And so that invading phage DNA is no longer functional, and then it can degrade that DNA in the cell. Now, this has more recently been utilized to edit genes and genomes in living organisms. It was first used in what we call knockouts, which is removing function from a gene. And so it was used to introduce mutations into a gene sequence. So in this system, the guide RNA, the RNA that recognizes a specific sequence, is actually engineered for the sequence that you want to edit. And it goes into a cell, recognizes it, and makes a cut. Now, this can be used in one of two ways. This can be used to sort of allow the cell to repair the cut and insert random nucleotides, which is going to disrupt the sequence or knock out the function of your gene. By doing that, you can study what that gene actually does in a cell. The other way that this is used is to actually edit or correct mutations. So if there's a known mutation, you can actually use the CRISPR-Cas system to go in, recognize the mutated DNA, cut it out, and then repair it with the correct sequence. So there's a lot of application for this. If you YouTube it, you can find all kinds of crazy information about designer babies and all sorts of things. It's actually been used quite a bit. There's been a lot of research on it. The first time it was used successfully was in 2014, and researchers were able to correct a genetic liver disease. It's a human liver disease called tyrosinemia. And they were actually able to restore function in enough cells that it was no longer fatal. They didn't cure it, but they were able to actually put in the correct sequence and make an almost normal life for some of these mice. So you can see there's a wide variety of applications, from correcting genetic disorders to cancers, all kinds of things All kinds of genes could be edited and repaired. You can also imagine that there's a lot of ethical questions in this. Who gets to repair their genes? Who gets to decide what genes get repaired? Who gets a say? Should we be doing this? Should we be doing this for fetuses and children and things like that? So there's a lot of issues with it, but it is a really cool technology, and there's a lot of things that can be done with it.